The learning objective for this first session, mine and David, is to be able to apply the social biological interface within a research context. So David gave you the theoretical framework to guide you to understand these mechanisms. So to achieve that, really don't want to rehearse too much of it, but just give you the illustration of the recap of what David just told you, and give you the move on to the cirrhosis of liver as an example. How we're going to apply the framework to understand this liver cirrhosis within the context of the heavy drinking, starting with the function of liver and the chronic alcohol misuse as a risk factors, and then how it is going to be, moving on to the processes, and what the sort of the objective measures we can use to assess this liver function, and to end with the summary. And this is fine. David's criticism came from the social determinants of the health model from the Bruno and the Marmot. Because like he said about all this exposure and processes, social, biological, kind of mixed together and assuming that's how it happens. So what he did in I hope David likes it, just make it a bit more clearer. And in this framework, he identified 3 exposures: structural, cultural, behavioral, interpersonal. This going through the biological process of material, CNS mediated, and epigenetics, and then affects the health. It's just a simple form of what he just talked about. But the principle of the interaction is in your handouts and your reading material. So I think if there any question about it, we can answer that. I think I have time for this. Then moving on to the social biological interface application, chronic heavy regular drinking and liver damage, i.e. the cirrhosis of liver. I don't know how much of the people know about human physiology and biology, but the liver, what the liver does is, in a nutshell, it's detoxing. What we eat, like drinking, sometimes the chemicals, the contain chemicals, liver does all this breaking down the harmful chemicals. Sometimes even the proteins that the byproduct of the breaking down protein, that causes the very harmful toxins for the body, but the liver does all this breaking down to make it unharmful again, so that we can safely excrete it through the urine. That's what the liver does. Then also what the liver does, the breaking down glucose, what we eat with the carbohydrates, breaking down to the it was breaking down the digestive system to making to the form of sugar. It's called glucose, but the liver does the breaking down glucose in a more further to make it as the glycogen and then stored in the liver. So that when we need immediate energy, liver can release, so that we can sort of use as an energy. Also, what the liver does is it produces bile. That's the sort of look like the juices, the very dark juices. But what bile does is the helping the digesting, breaking down the fat. So if the liver doesn't function, we cannot digest the fat as well. Also, more importantly, they also produce the broad protein, the proteins. And also macrophages that part of the cells that they fighting infections. So liver in one organ, but that's very multitasked. So important vital organ. So we have to look after. That's the thing. And cirrhosis, in terms of cirrhosis, what cirrhosis means is the scarred liver cells. due to chronic damage to the liver. So the liver usually causes silent organ because the liver really doesn't harm by the drinking straight away. It takes ages to the causing the liver to be damaged. I just want you to look, it may not be showing very well, but normal liver is very smooth. But when the cirrhosis, the scar tissue, it's kind of showing some irregular shape on the surface, so it's very looked like the irregular shape of the liver. And that's I don't know if you can see it, but it's the inside of the cirrhosis of liver. It's the very inside is quite damaged. It really doesn't look like the normal cells at all. For the cirrhosis, other than the drinking, but that's the talk with the chemical exposure. But for example, the alcohol is the one of the causes. But other than that, infection like hepatitis C or having the excess fat, it's called NASH, and the other factors, but some which are the mostly preventable. But just keep it back on your mind that it's not only alcohol causes a cirrhosis. And the alcoholic liver disease attributed by the heavy alcohol use is a major cause of alcohol-related deaths. And the ONS they show that highest in the age group of 50 to 59, they dying from the alcohol liver diseases. And what now that we going over, they just gave you some illustration of what liver does. So now moving back to our business about exposures. Exposure is mostly you can think of the heavy alcohol use that's this enhanced by the peer, like parents and what other people do, and so we kind of adapting the behavior. That's the one big exposure. But then I like to point out the job demands the insecurity. That's interpersonal. It's also the exposure. And that's the two examples I'm going to move forward. But also we can think about like the recent issues about alcohol pricing and accessibility to shops or outlets, or sort of they can be in the macro level we can look into as the social structural exposure. But I'm not, for the sake of this exercise, I'm not going to talk about it, but that's something you can take it back to your home and thinking that if you're interested, you can develop the hypothesis under this framework. And the interpersonal exposures, the Sieglitz and the others, they are talking about the psychosocial stress at work model, which fits into this interpersonal exposures. Like effort reward imbalances have a link with the heavy alcohol use, and also the decision making process. Also the associates with the heavy drinking men and the women. But why the people in a stress they may want to drink? That goes to the another the slides that we explain furthermore. This is we talked about interpersonal exposure. Then now we're moving on to the process, how the people interpret this stress, why the people may want to drink more. It's based on the stress model, the proof said the coping to drink. That's the theoretical approach. If the people the stressor, the induced by stress tends to drink the heavy drinking, you observe in the terms of the coping behaviors, like you want to avoid all the stress, and also the positive expectancy, that is the perception of the alcohol. If you're expecting to drink alcohol to be behaving in a way of the relieving stress or the feeling better, and the people tend to drink more. And in our the study, ICLS study, worked with the looking at the BCS 7 cohort data, and we have the measurement of positive expectancy in age 16. When we looked at this positive expectancy and age 30 drinking behavior, among men, with this, the presence of positive expectancy and the depressive symptoms at age 30, and together these men drink the heavily drinking more. So it's just the alcohol positive expectancy, the thinking if you're depressed, you want to feel better, and you tend to these people tend likely to drink more. And also, this drinking type of motives, found to be this abstract children study, found to be the more with socioeconomic status, the pattern, like the children from high school, high socioeconomic status, drinking is a part of gaining a confidence, but the people from the children from low socioeconomic status, drinking is the way of the coping with the low moods, so it's the supports the going back to this CNS mediated process of coping to drink. So the material process of the alcohol misuse and the damage to the liver, really, like I said, the liver being a silent organ, it takes ages to develop this stage. So there is the first stage, the second stage, and third stage to the finally entering into the cirrhosis of liver. The first stage of the liver, the damage is just the accumulation of excess fat, and the second stage is going to the moving to the called alcohol hepatitis, and observed by swollen liver. There's some the death of the cells occurring. The third stage is we call the alcohol cirrhosis, permanent scarred liver tissues. So that's fine. That's something that I showed you. That's the image from here to there. It really takes long time. So I think the template in here for the tomorrows. So I developed all this diagram of how this might be, just what I said, how this moving on to each other. So they are high job demands and high job insecurity, and they are causing a stress and interpret the CNS mediated process, and they wanted to drink to cope this stress, and then moving into they learn the way of the drinking, and then going to the material process of the liver damages, and they are causing to the impaired liver function. But they also study suggests that this process really was accelerated by the pre-existing obesity. So that's really the bit more than complicated picture. But if you want to tackle it, you can add it to the obesity into the model and further expanding to it. That's fine by me, and we can help that. And if they like developing this possible research questions under this my example of the frame drawing, then what I usually like to do is by the arrow to arrow, how the association would be existing. So they would be like the high job demands or insecurity related to heavier alcohol use, or how long-term the unemployment rate, long-term and heavy alcohol use. Would high positive alcohol expectancy explain the relationship between the high job demands and unemployment or heavy alcohol use and liver function? That's some mediating role of CNS part would be explained all this exposure to the outcome. I usually take that sort of approach, but the other people may have different approach to answer the research question. That's perfectly fine, but that's just only example. So what the biomarkers that we really can use to show this the alcohol use and to the liver function, the Altaro et al. In 2009 and show that this the ethanol intake is strongly correlated with GGT, AST, ALT, and ferritin. Ferritin is the indication of the iron in the blood. But also they said that the upper normal limits, if you're talking about all these biomarkers, the cutting off the normal and abnormal, the normal limits is also just little bit different by the gender, also the drinking status. If sometimes the most bit higher if you're drinking moderate, drinking heavy, moderate drinker compared to us the people who never drank. So that's something that you have to look into the literature when you want to draw the cut line. That you have to take it into consideration, depending on your biological makeup and also other external factors. And also, if you're talking about the alcohol cirrhosis, there is the established scoring methods, including the bilirubin, creatine, prothrombin time. So that if you really want to look at the alcohol hepatitis and the cirrhosis, there is some. This is the reference that I brought it with me. So if you want to look at the paper, the more than welcome that I can show to you. So there is another way of the clinical methods to indicate the alcohol hepatitis and the cirrhosis of liver. Repeating myself with this previous slide, the GGT, AST, ALT, also if it's possible, platelet counts and protein time, the serum albumin, is the protein, usually the protein in the blood, but the protein is so important. Is also they are showing the liver function. It's very highly connected associated with the liver function. So that's something we can, in addition to this enzyme, we may want to look into albumin as well. And then also then I put in waste, at the end of the stage of the liver cirrhosis, people are having a huge waste because they have the water and because of the liver dysfunction, that they tend to contain the water in around the tummy, so it has a huge waste circumferences. So something may not be seen that the normal circumstances that might be possible. But other than that, the objective, we also need to look into subjective measurement, like the general feeling of unwellness, indicated by anorexia, weakness, fatigue, weight loss. Sometimes they see the bruises and tendency to bleed. And some people say that in the medical condition question that I have the cirrhosis or I'm suffering from hepatitis. And also if you have the opportunity to be able to use administrative data link, you can also use the hospital data information to validate all these conditions. And it comes to the mind ELSA, English longitudinal study of aging on White Hall. They have repeated measures of biomarkers and also subjective measures. That's really wonderful data set that you can apply to the measure the repeat process of disease, ongoing disease process. Especially the ELSA is the opening available by UK Data Archive. That's something that you may want to think about using it, but although the people, the sample size may be a bit smaller toward the end of it, but that's good. So I really like to say that we talking about the biomarkers, but the biomarkers, they're not diagnostic tools. So we really have to see all the variables in a comprehensive manner, and sometimes subjective ways of complaints can also able to interpret all these findings. So that's just keep it in mind. Like I said, the cirrhosis can be caused by the infection as well. So just to summarize, we have, as a researcher, we have a challenge to understand socio-biological pathway that you applying to theoretical framework by the understanding of what are exposures and what are biological processes, and what David talked about the governing principles, how these processes can interact and accumulate over time. And the glossary of the biomarkers, along with understanding society, when we send it out in our glossary, we have the biomarkers, explain what do they do,